Hello everyone, and a warm welcome to this webinar on the topic developing vaccines against outbreak pathogens. This webinar is part of a series of planned webinars with the goal of providing information on available career options in the field of vaccinology. My name is Clive Nyaribo, Assistant Research Officer at KEMRI-Wellcome Research Program, providing support of the Vaccine Workforce Development Program. Together with my colleagues, Professor George Rimwe, Marianne Munene, and Ravendra Nafura, we welcome you to this session. Before we get started, a few housekeeping matters. Kindly take note that this webinar is being recorded. Kindly feel free to use the chat option to share questions during the presentation, and we will address them during the Q&A session. We'll have a few open slots for the attendees to directly ask questions to the presenter. If your internet is stable, you can raise your hand using the available icon. As earlier mentioned, these are a series of planned webinars with the goal of providing information on available career options in the field of vaccinology. This being run under the KEMRI-Wellcome Trust Research Program, which is a well-known health research unit of excellence based in Kilifi and Nairobi in Kenya, as well as Mbari in Uganda. Over the years, we have excelled in the use of novel ideas and collaborations to achieve better health for Africa, while also developing African scientific leaders. Our vision of the program is to undertake cutting-edge and novel research relevant to national, regional, and global needs. It's under this program that we have the Vaccinology Vaccine Workforce Development Program that aims to expand the technical know-how and capability for sustainable end-to-end vaccine research and development in Africa through the training and knowledge exchange activities in the vaccine ecosystem. This involves areas such as target product profiling, research and discovery, preclinical and clinical studies, biomanufacturing, regulatory affairs, and deployment. The workforce development is through 4 channels. We have one, internships at vaccine manufacturing institutions that aim at establishing a mentorship scheme that links interns with senior vaccinologists and LT stock market systems, and engagement with academics globally to share and strengthen experiences in vaccine R&D and manufacture and enhance career development. 2, we have a full funded master's level course in vaccinology, where applicants are selected based on their direct involvement in vaccine research and their professional excellence and academic achievement. And 3, we have webinars like this one today to other career scientists, graduate and undergraduate university students in Africa to provide information on possible career options for university students for informed career decision making. 4, we also have the Vaccinology School Engagement Program, where students from local secondary schools are invited for lab tours and career talks to spark their interest in science, especially in the vaccinology field. This program is in alignment with the African Union's platform for harmonized African health products manufacturing, ambition to increase local vaccine production to meet 6% of Africa's routine immunization needs by 2040, and strengthen the capability for vaccine research, discovery, and manufacture in Africa. And this webinar series is part of this engagement. Today, we are glad to have Daniel, who will be discussing the topic, developing vaccines against outbreak pathogens. Mr. Daniel Wright is a senior researcher and lab team co-lead at the Oxford Vaccine Group and Pandemic Science Institute, working under Professor Teresa Rampe, OPE, to develop vaccines for emerging viral pathogens. He is Doctor of Philosophy in Clinical Medicine, focused on a One Health mold species Rift Valley fever vaccine, and was based between Oxford and Kilifi, Kenya. He continues to work on the JEDOX RVF program with Professor George Warime and lead multiple other programs, developing Rift Valley fever and new world arenavirus vaccines. Over to you, Danny. Thank you.
Thanks, Clive. A nice introduction. Hi, everyone. Hope everyone can hear me. So I'm going to talk about developing vaccines for outbreak pathogens. And what I mean when I talk about outbreak pathogens, these are the sorts of pathogens that are capable of causing sort of large outbreaks, explosive outbreaks, large epidemics, potentially pandemics. And really the whole landscape of research into countermeasures against outbreak pathogens really changed in 2013, where you may remember a very large Ebola outbreak that was occurring in West Africa. And up until that moment, Ebola was a horrific disease, really high case fatality. But in terms of burden of disease, it was relatively small. The outbreaks were relatively small, and they petered out on their own without direct use of vaccines. And that was the case, lots of little outbreaks, relatively little outbreaks, until 2013, when there was this explosive outbreak. And suddenly what was previously a disease that was causing these small outbreaks exploded into a disease which was killing thousands of people over several years. The outbreak lasted. And there was ongoing research into trying to develop Ebola vaccines up until that point. It was obviously a need, but given the relatively low sort of public health danger from Ebola, there was a lack of, I guess, a lack of funding, a lack of urgency to really progress these vaccines before it became a huge public health concern. And so after this very large outbreak of Ebola in 2013, it was considered a bit ridiculous that, now it's become a huge problem. We desperately need vaccines, and there was a mad scramble to try and make and test vaccines against Ebola. And it was considered that this isn't a reasonable way of doing things. We don't need to wait until these diseases become a much larger problem, where we think, okay, now it's worth developing a vaccine for. We need to really prioritize research and develop these vaccines before they become a huge problem, so that instead of just responding to these large outbreaks, we can actually do something about them and prevent them from becoming much larger. So after 2013, international organizations like the WHO, the African Union, Coalition of Epidemic Preparedness, and others, they periodically release a list of what they consider priority pathogens of concern. And these are sort of outbreak pathogens where we either don't have vaccines or we don't have good enough vaccines, and we really need to focus on making them so that some of these diseases already have a significant burden of disease. Some of them are relatively less so, but they all have the potential to be much worse. And so these on these lists are things like CCHF at the top, so Crimean Congo hemorrhagic fever, Ebola, Marburg, everyone's heard of, Lassa fever, big problem in West Africa, MERS and SARS, and everyone knows what happened when we had a new SARS coronavirus a few years ago. Things like Nipah, Rift Valley fever, which I'll talk about later, Zika. And then there's also disease X. And disease X is a recognition that whatever causes the next large outbreak or potentially pandemic, it's not necessarily going to be from one of these diseases which we know about and we know has the potential to be much worse. It may be something completely novel. So it might be something like SARS-CoV-2 was back in 2019. We didn't know about it as a pathogen. So it's a recognition that we need to develop vaccines against the diseases we know about, but we also need to develop vaccines, or we need to develop the capacity to quickly respond to outbreaks so that we're able to develop vaccines against diseases which at the moment we don't know about. So for this webinar, I'm going to cover a sort of whistle-stop tour of a few different vaccine programs that we're involved with. So everyone knows of Ebola virus and SARS-CoV-2 during the pandemic. I'll introduce the others, the Rift Valley fever, if you're less familiar, and Junin is a viral hemorrhagic fever in Argentina, which is also probably less well known. So before we go into the individual programs, I just want to say a little bit about vaccine platforms. And what we mean with vaccine platforms are a sort of method of manufacturing a vaccine which doesn't change regardless of what the vaccine is actually targeting. And so two examples of vaccine platforms, and there are many others, these are just two examples. An adenovirus vaccine platform, there are mRNA vaccine platforms, and they're vaccine platforms because the technology and the manufacture of them, it doesn't change. Whether you make a vaccine against HIV or malaria or TB or Rift Valley fever, the only bit that changes is when you decide which part of that pathogen, the gene from which part of a particular pathogen is going to be encoded in your adenovirus, for example. That's the only bit that changed it from an HIV vaccine to a malaria vaccine. The rest of the manufacturing process, the way that we clone the gene in, the way that we manufacture the adenovirus, the reactogenicity that adenovirus is called, that is all pretty much agnostic of the gene that you've put in. And it's the same with mRNA. So the way that you make your mRNA from your cDNA, the way you incorporate that into the LNP and the formulation of your LNP, that's already decided. And so you can very quickly adapt your adenovirus platform or your mRNA platform or whatever other platform you have for a novel vaccine. And so these are particularly useful for outbreak pathogens where, particularly if it's a new pathogen and people have just identified that this new disease is spreading, we need a vaccine against it. These sorts of vaccines are very beneficial because they're so quick and they're so easily adaptable. And it's not a case of, for example, reinventing the wheel and trying to say, okay, we want a live attenuated vaccine. And there are many licensed, incredibly useful live attenuated vaccines. It's not that these are the only two good platforms. It's not the case at all. There are a host of different vaccine technologies, all of which have advantages and disadvantages. But when it comes to being quick and responding to outbreaks, having a way of making that vaccine quickly is very beneficial, rather than something that's more bespoke and saying, right, we need to attenuate this virus and then trying to understand how best to do that, and it changes depending on what the pathogen is. These vaccine platforms remain the same, and it's just a very quick way of making new vaccines. So the vaccine programs that I'm going to talk about, they do focus on adenovirus and mRNA, but I do want to make clear that these are not it's not the future is just adenovirus or mRNA. It's all of the vaccines that have been developed, proteins, live attenuated, inactivated. They all have a very important role to play in different diseases. So the other sort of concept I want to mention is this idea of a One Health approach to vaccinology. You've probably heard of the term One Health. It's often somewhat vague, but essentially what I mean is taking a holistic approach to vaccinology. So taking expertise from the veterinary side, from the clinical side, understanding that in order to successfully roll out a vaccine and actually improve public health, it relies on all the other expertise and the other sort of areas. You need better surveillance, you need better diagnostics. And this is, again, particularly relevant for outbreak pathogens because the vast majority of new diseases originate in animals. And when animal outbreaks do occur, particularly if they involve livestock, there is a huge economic impact. So you have the death and disease from the livestock, which can then reduce the herd size, which then causes reductions in production. And then that can last generations until the herd size is rebuilt. And then there are trade embargoes, which can be catastrophic for economies that are particularly reliant on livestock trade. There are also reductions in food security. And both of these things, while we're just talking about animal outbreaks, or livestock outbreaks in this case, they ultimately lead to worse human health. And then on top of that, when you have these animal outbreaks, you increase the risk of spillover into humans, where we directly get affected by the disease. And so all of these three things ultimately lead to worse human health. And if we can protect animals, then it helps protect humans both directly by reducing the chances of spillover, and also indirectly by reducing or limiting the negative consequences of animal outbreaks. And so that brings us on to the first vaccine program I want to talk about. This is Rift Valley fever virus, which is a mosquito-borne disease. It's spread by numerous different species of Aedes and Culex mosquitoes, and it's largely a disease of livestock. So it affects sheep, goats, cattle, and camels. And it causes all sorts of clinical signs in animals, but the two major ones are really high rates of neonatal mortality and really high rates of abortion. So often the first signs of an RVF outbreak is when you get kind of simultaneous abortions in the herd. It's called an abortion storm. And then that's one of the sort of key indicators that there's an RVF outbreak on your hands. And so just livestock outbreaks, forgetting all about the human side, has a significant economic impact. As you can imagine, if your animals are dying, if it's causing abortions, that themselves or in itself is a significant problem. But it also spills over into humans, and humans become infected either through mosquito bite, like the animals, or through contact with infected animals. So it's people that work with animals, particularly people that are involved in their slaughter or other animal husbandry, that are most likely to get RVF infection. And when we become infected, there's a wide spectrum of clinical outcomes, from self-limiting febrile illness on the mild side, all the way through to hemorrhagic fever, encephalitis, and a very high case fatality rate. And so typically you'll see case fatality rates for RVF that are sort of maybe 30% plus, and that's usually it's, I guess, in some ways inflated by the fact that people who get diagnosed with RVF are the people that have severe illness and seek hospital treatment. The people on the milder side tend to not seek hospital treatment. And so the actual case fatality is considered more like sort of around 1-2% perhaps, which is still very high. But there's a lot of people that will get more mild disease, and when you do see an outbreak, it's usually a massive underreporting of actually how many cases you've got. And on top of humans, there's a whole host of wildlife species that also become infected. And so in terms of how do you reduce the burden of RVF disease, we need better livestock vaccines. We do have them, but they tend to be live attenuated, which are contraindicated for use in pregnant animals. So RVF replicates very efficiently in placental tissue, and the vaccines themselves that are live attenuated can cause teratogenic effects and abortions. So usually it's pregnant animals that are not able to be protected by vaccination. On the human side, we have no vaccines at all, no licensed vaccines. So we're in desperate need of a human vaccine. On the wildlife side, we don't really know much about the burden of disease in the different species, but that is to say, because it's so promiscuous and it infects such a wide range of species, it's not a disease which we're going to be able to eradicate. We can, our best hope is to have good livestock vaccines and good human vaccines. So this vaccine is the program led by Professor George Grimway, and been working with him on this for a number of years. This is, I suppose at the top is the kind of general vaccine development pipeline. So it starts on the left-hand side with vaccine design. People have made RVF or tried to make RVF vaccines before. They've tried live attenuated vaccines, which they have their strengths. They can be very immunogenic. The drawback is that it's there are safety concerns with reversion to virulence, or certainly if they're ever to be used in pregnant women, which would be some of the people in need of protection. And there have been inactivated vaccines which also suffer from the same, I guess, advantages and disadvantages that inactivated vaccines have. They're very safe, but they require multiple doses in order to get a protective immune response. So we wanted to do something slightly different. We didn't want a live attenuated or an inactivated vaccine, so we went with a viral vectored vaccine. This is the surface of Rift Valley fever, and there are two glycoproteins, one called GN, one called GC. They form these heterodimers, and then those are arranged into these ring-like structures, and that's what covers the surface. And we know for Rift Valley fever, what we really want to do is develop neutralizing antibodies, and we know that neutralizing antibodies target both GN and GC. In terms of why we want to develop neutralizing antibodies, there's a lot of evidence. There isn't a sort of a bona fide correlate of protection that says you need this many in order to be protected, but it's very strongly associated with protection. So you can challenge an animal, and then if they're a convalescent animal afterwards, then you take the serum, you passively transfer it into a naive animal, and you get complete protection. And it's almost unheard of to see detectable neutralizing antibodies and not see complete protection. And it also seems that if you become infected once, then you're protected for potentially for lifelong, and potentially for at least a very long time, so decades. So what we really want to do as a vaccine program is to make a vaccine which is capable of inducing neutralizing antibodies which target GN and GC, and are therefore protective. So we take the genes for GN and GC, which is an RNA virus, convert it into DNA, incorporate it into the chimpanzee adenovirus. So this is ChAdOx1. This is essentially exactly the same as the Oxford-AstraZeneca COVID vaccine, except instead of encoding the spike protein from SARS-CoV-2, it encodes GN and GC, which is a sort of polyprotein, which is then post-translation cleaved into the separate GN and GC. And so the idea is that when you vaccinate, perhaps this gets taken up inside a muscle cell, and then this insert here will start expressing, and the virus will do what viruses normally do. They'll hijack the cellular machinery, and your own cells will start producing the Rift Valley fever GN and GC proteins for your immune system to see. And so after initial, so usually the first step after you've made the vaccine is you test these vaccines in small animals like mice to get an initial idea of whether it's immunogenic, potentially whether it's protective. For Rift Valley fever, we wanted this vaccine to be used as a veterinary vaccine and also a clinical vaccine for humans. So the idea was a single vaccine that could be used in the multiple livestock species and humans. And so we went into the livestock to see, can this vaccine protect the target species as a veterinary vaccine? So that's ChAdOx1-RVF. RiftVax is the licensed vaccine. It's the most widely used one. It's a live attenuated one, as I mentioned, and it causes abortions in pregnant animals. So it's considered very effective, but it's not used in pregnant animals. And then PBS is the sort of placebo. So we took sheep, goats, and cattle, and we vaccinated at day 0 with a single dose. And then 4 weeks later, in order to see whether this vaccine was efficacious, the endpoint was we challenged them with live RVF, and then every day for the next 2 weeks, we look in the serum and try and measure RVF viremia by RT-PCR. And we found that actually there was complete protection in all animals that were vaccinated with 1 dose of ChAdOx1-RVF. RiftVax, we saw complete protection in goats, but a single sheep and a single cow failed to seroconvert post-vaccine and then subsequently became viremic after the challenge. So even though these numbers were relatively small, we saw complete protection with the vaccine, and we saw that it even outperformed the licensed livestock vaccine, which was good news. So I mentioned before about the need to protect pregnant animals. This is something that the current livestock vaccines, at least the most widely used one, is not capable of. And so we wanted to see, can this vaccine, firstly, is it safe in pregnant animals? And secondly, does it actually protect from RVF? And typically, if you're not vaccinated and you're a pregnant sheep, then your chances of an abortion are sort of 90 to 100%. We took 8 animals at the start of their second trimester in each species, or 16, half were vaccinated, half placebo. Vaccinated again, single dose, just at the start of the second trimester. Then they were challenged 3 weeks later, and then we followed up for any signs of disease until the end of the study, when the autopsy and the fetuses were looked at. And we found these little graphs in the bottom. This is viremia post-challenge, where the blue is placebo animals, where viremia peaks after a few days, and then red is the vaccinated animals, where we see no viremia at all. And so the high-level results were that 100% of all the vaccinated pregnant adults showed no signs of disease. And in sheep, in the 8 vaccinated sheep, these were all multi-fetus pregnancies. There were 18 healthy fetuses, and all of them appeared healthy. So complete protection in sheep. In goats, there were 2 fetuses from 2 separate goats that did seem to succumb post-challenge, but 23 out of 25 survived. So this was great in that we've shown that this vaccine, compared to the sort of the competitor licensed vaccine, which can't be given to pregnant animals, not only is it safe to give this vaccine, or it was safe in this study, but we showed protection against challenge, sorry. And so this study did end a few weeks after the challenge. Further down the line, we'd look to do a study where the animals are brought to term, then we can show conclusively that they give birth to healthy animals. But at the first step, this is very encouraging that we can see the vaccine is highly protective against disease and fetal loss. So on the veterinary side, it looks very good, but we also want this vaccine to be protective in humans. So we've done 2 phase 1 clinical trials, one in the UK and one in Uganda, with relatively small numbers, as phase 1s tend to be. 18 to 50-year-olds, broadly healthy, and it's a dose escalation trial. So we started at a very small dose, and then we end at the dose we would intend to give. And this is the dose, the 5 times 10 to the 10 viral particles that the COVID vaccine was ultimately rolled out. So that's the dose that we would ultimately expect to give. And it was a single dose study, so one prime vaccine at day 0, and then taking blood, PBMC and serum at multiple time points for the 3-month duration. And I would show all the immunogenicity data, but this is looking at the key sort of neutralizing antibody titers at day 28. So it's typically the peak of response, about a month after the vaccine for the 3 dose groups. In the 2 different trials, so the UK trial in blue, the gold squares for the Ugandan trial. And we don't see any real difference between the UK and Uganda, where RBF is actually endemic, which is good to see. And crucially, we have another comparator where we've looked at naturally acquired infection. And for Rift Valley fever, we know that naturally acquired infection is very good. So people who are infected, they get neutralizing antibodies, and those neutralizing antibodies last for seemingly for decades. And based on animal studies, we think that if you have even a whiff of neutralizing antibodies, you're very likely to be protected. So we think this is a protective response, and we can see, although the due mean is a bit lower, we can see that it overlaps very well. So it's good that we're seeing an immunogenic response, particularly in the top dose group where all the participants are responding, and that overlaps strongly with what we believe to be a protective level of neutralizing antibodies. So in terms of that vaccine program, we are essentially here. So we've designed the vaccine. There's been several preclinical studies. GMP is sort of the standard of making vaccines for human use. It's much more expensive to make clinical grade vaccines. That's been done for 2 phase 1s. There is a phase 2 ongoing in Kenya now, which has finished enrollment and is in the sort of follow-up period. But to go from here to here has taken years. And that's not abnormal. It's generally how long it takes. Years and years. Started over 10 years ago to go from when George initially made some vaccines to we're halfway through a phase 1, phase 2. Now, what tends to happen with working on vaccines against outbreak pathogens is that the outbreaks don't necessarily stop. They don't wait till you finished your vaccine program. So there was a comparatively large Rift Valley fever outbreak recently at the end of last year. And that sped some things up. So because of this data that we had on the veterinary side, and importantly, we had some clinical data, it was one of the leading candidates to be rolled out to test efficacy should this outbreak in Senegal, Mauritania, and the Gambia continue. So when outbreaks do occur, being in a position where you already have at least phase 1 data is a huge advantage. And it means that when an outbreak does occur, you're in a better position to try and test efficacy of your vaccine. Because one of the biggest challenges with demonstrating efficacy for outbreak pathogens is that it's very difficult to do, because in order to test efficacy, you need to go somewhere where there's a consistently high burden of disease, where you can do a trial there and say, well, vaccinate half the people, and then the other people will receive placebo, and then we'll count the number of cases. But for a disease like Rift Valley fever and for other outbreak pathogens, actually there's nowhere in the world that you can go and see a consistently high burden of disease. So how do you actually test efficacy? And one of the ways is you be in a position where you have vaccine available so that you're able to test it should a large outbreak occur. But it's very difficult, and you need to really, really be quick, because a lot of these outbreaks, like the Ebola outbreaks previously, they peter out on their own, which is great, or that they have done at least previously. But obviously that leaves a wake of a huge number of lives lost. So if you're in a position to test efficacy quickly, when the outbreaks do occur, then at least you can learn something. You can test your vaccine. So in the absence of an ongoing large outbreak, how do you license these vaccines? And one of the ways is via what's called the animal rule. So when it's not feasible to do phase 3 efficacy testing, either because there aren't any ongoing outbreaks, there aren't large enough ongoing outbreaks, or for ethical reasons, if it's not feasible, you can do an efficacy study in relevant animal models, so typically non-human primates, and you can measure the immune response in those animals that are protective. and then you can compare it to humans. So you still do essentially a phase 3 human study, but you don't look at efficacy because for whatever reason you can't. You still need thousands of people so that you can generate safety data, and you can look at the immunogenicity, and then you can compare the immunogenicity in animals that you know are protected, and then bridge that to humans and say, well, this is what the immune response looks like in protected animals. This is what the immune response looks like in vaccinated humans that we don't know are protected. But given, for example, let's say we know neutralizing antibodies are crucial, that's the correlate of protection. This amount is protecting animals. This is the amount we're seeing in humans. And essentially you're trying to build a case to regulators to convince them that the immune response that your vaccine is generating in humans is equal or better than the protective immune response that you're seeing in animals. And it's not just a straight comparison of certain immune parameters like neutralizing antibodies. It involves knowledge of correlates of protection. What is it that's actually doing the protection? And so there's a lot of sort of immunobridging, and it's not necessarily it's not an easy way to do it. But vaccines have been licensed via the animal rule when efficacy testing either wasn't available. So that's one of the routes that you can go down to actually be in a position to have a licensed vaccine prior to a large outbreak occurring, so that you're ready when one does occur. So that's it for Rift. Just a couple of slides on Ebola. So as I mentioned previously, this kind of changed the landscape back in 2013 when there was a comparatively huge outbreak of Ebola. And that was caused by Zaire. There are 4 different strains known to cause human disease. As a disease, it's spread by bats, and bats can infect other animals. And then humans become infected when they come into contact with these animals, and that can be via bushmeat, for example. And then human-to-human spread can occur, through unprotected contact with blood and bodily fluids. And this was the outbreak that I was talking about earlier in 2013. I think the final number was something like 11,000, and even that was considered potentially a significant undercount. So really, if we were in a position to test these vaccines that had already been shown efficacious in NHPs and really prioritized developing those vaccines and running clinical trials prior to this outbreak, then we would have been in a much better position. So this outbreak actually lasted a few years, and I just want to briefly mention the only vaccine that actually was able to demonstrate efficacy during this outbreak. So something good came out of it, in that at least it didn't peter out. It did peter out on its own, but it didn't peter out without us gaining any sort of knowledge about whether these vaccines that were in development worked. So this was a VSD vaccine, so another viral vector vaccine, and it contained the glycoprotein for the Zaire Ebola. And how they tested efficacy, which came towards the end of this outbreak, I've taken a picture from a separate SARS-CoV-2 paper, but just to illustrate this sort of ring vaccination approach, where they identify a case, and then they identify the contacts of that case, and then they put a ring around all the contacts of that first exposure, and all of these different rings form these different clusters. And essentially they vaccinated each of these rings with this vaccine either immediately, or it wasn't considered ethical to use a placebo during the outbreak, so they either vaccinated them immediately or after, I think, 28 days. And then the comparison was, do rings that were vaccinated immediately show fewer cases than the rings that were vaccinated after 28 days? And it did. It showed that there were actually no cases after 10 or so days after vaccine. If they were vaccinated immediately, no cases compared to 20 or so cases in people that were vaccinated later. And it was later sort of confirmed to be highly efficacious. And so it was great that this large outbreak was able to be used to ultimately demonstrate efficacy, but thousands of people died. If we were just a bit quicker and we could, if this trial with this vaccine was ready to go even earlier, then thousands of lives could have been saved. So that's sort of the aim going forward is just to be quicker, because speed has such a huge impact on the public health burden of outbreaks. So there are licensed vaccines now against Zaire. There are not licensed vaccines against the other the other species of Ebola. So there's been various outbreaks of Sudan virus recently. So Professor Teresa Lam has several vaccine programs on filoviruses, and really the ultimate aim would be to generate a vaccine which is capable of protecting against multiple different species or multiple filoviruses, including Marburg. And if we can get a position to test these now, then we're in a much better position. That certainly, if we get to at least phase 1 clinical data with these new vaccine candidates, that should an outbreak occur, we're in a quicker position to try and test them and use what these outbreaks are not going to stop. They're going to continue. So as bad as they are, at least we want to be in a position where we can test vaccines when there are no vaccines available. Because if an outbreak occurs and then it peters out, then you've still got the same disease burden, but you've not learned anything. Whereas if we're in a position to actually test our vaccines, then even if they don't work, at least we can go back to the drawing board with something concrete and say, well, this vaccine produced this sort of immune response and it didn't work. So we need to go back and try and do something differently. So moving on to something which is everyone is very familiar with, COVID. So this is developing a vaccine, not just for an outbreak pathogen, but in the context of an ongoing pandemic. And so when people started becoming sick in China at the end of 2019, it was relatively quickly identified as a novel coronavirus. And so what we did as sort of vaccine developers and the various people that made vaccines was identify what part of the pathogen we think is most important. So that's typically the receptor. So the part of the pathogen which facilitates infection, essentially. If you can generate antibodies, for example, that bind that and prevent infection, then you can prevent infection, you can prevent all the downstream more severe effects. And so we took the spike protein, as did pretty much everyone else who was developing vaccines. We put it into an adenovirus in exactly the same way as we had made Rift Valley fever, Ebola vaccines, and previous vaccines before. So it is very quick in that respect. Once we've identified the gene, or the gene's been identified, then it's very quick to make an adenovirus in exactly the same way as we had previously made it. And then essentially the adenovirus is a sort of Trojan horse. So the actual adenovirus is a vehicle to essentially deliver the spike gene into people's bodies so that their own cells can start producing a spike protein and then generate an immune response. And it was the same for some of the larger mRNA companies that were developing vaccines. They identify the spike protein and they use exactly the same process as they had previously used for developing mRNA vaccines, and it's very quick. And so this is, again, the typical sort of development process and timeline of developing vaccines. And it's generally done in a linear fashion. So you do your preclinical studies, and if that looks good, then you move on, you do your phase 1, etc. So what happens during a pandemic is that the burden of disease and the sort of economic cost of the pandemic is so large that you can't wait this long. This can easily take 10, 15 plus years, and that's when things are going quite well and you've got a vaccine that actually works. So it turns out that actually you can go much quicker. And there are a variety of different reasons why COVID vaccines were able to be developed so quickly compared to previously. But one of the most important parts was taking economic risk, and it's only economic risk, by pushing these steps in parallel. So all of the steps are still done, but instead of waiting until you get the data, for example, from the preclinical studies and thinking, oh, that looks good, then applying for money and then making the expensive GMP grade vaccine for a clinical trial, actually you're starting the preclinical studies and shortly after you're straight away making GMP vaccine in case it looks good. And then when you get the data from the preclinical studies that it looks good and you can go ahead, then you start your phase 1. But at the same time as you're starting your phase 1, you're pouring hundreds of millions into producing enough GMP grade vaccine for a phase 3. And then shortly after that, you're upscaling, you're producing hundreds of millions of doses of this vaccine, all before we've got any idea if it actually works. And so typically, that's not how it works, because people don't want to spend an enormous amount of money on something which might not work. Because if we've made this vaccine, we've produced hundreds of millions of doses, and then it turns out that in phase 3 it shows that it's rubbish and doesn't work, then they all go in the bin. But if you're in a pandemic and it's costing the sort of global economy trillions and it's shutting down the world, then if you want to go quickly, you've got no option but to take economic risk, because the risk of not doing anything or not having a vaccine is even greater. So that's largely how the vaccines were developed so quickly. There was no reduction in safety data. The trials are still very large and very well powered. It was just that there was economic risk by doing a lot of the steps in parallel. And so one of the dangers or the concerns around developing SARS-CoV-2 vaccines were that back with SARS-1, there was some of the vaccines that were in development showed a phenomenon called vaccine-enhanced disease, where the immune response that they elicit was so unhelpful for SARS-1 that when animals were then challenged with SARS-1, if they were vaccinated, they got more severe disease than if they weren't vaccinated. Now that's sort of worst case scenario. We think we're developing a vaccine, worst case is it doesn't work. Well, technically the worst case is it actually makes things worse. So before we rolled out our clinical trials, we wanted to be sure that the vaccine that we developed is not going to make things worse, because this was a pandemic. People were, pretty much everyone was likely to get COVID at some point. And so if everyone vaccinated in our trial was therefore more likely to get severe disease, that would be pretty catastrophic. So we did a study in a variety of different preclinical animal models. This was in NHPs, showing that actually the vaccinated animals did massively reduce viremia post-challenge, and that their lungs were all histologically normal, and the vaccine did prevent pneumonia. So not only did it not cause vaccine-enhanced disease, it did provide protection. So that was the data that we needed, at least the data that it wasn't going to make things worse, so that we could then go into phase 1 clinical trials, which were carried out in a number of different countries: South Africa, Kenya, the UK, Brazil. And really, from design of the vaccine to approval was only 11 months. So I'm talking about how the Rift Valley fever vaccine, which is going very well, it's taken years, and that's fairly normal. Whereas this, we started in January, and we had vaccines by the end of that year. And it was really split into sort of three parts. The first was making the vaccine, the next, and then doing the preclinical studies. Then it was setting up all of the clinical trials. And then the last part was really just waiting for the cases to accumulate. So that takes time, specifically especially in the context of lockdowns, where maybe they were important and needed to reduce the spread of disease. But in terms of finding out if your vaccine works or not, I don't want to say you want as much disease as possible, because obviously you don't, but you will get an efficacy readout quicker if if there's more disease around. So in terms of real world data, this is just data from the UK, but it really shows how powerful these vaccines were at preventing deaths. So on the left here is in red is what was the second wave in the UK, was in the pre-vaccine era. So we had number of cases down here, and then the associated number of deaths over here. And then after vaccines, in the next wave, when they were introduced and rolled out, we actually got a similar number of cases, at least because as the virus started evolving, they became better at causing infection and evading the vaccines in terms of being able to infect. But the vaccines and the various ones that were rolled out were still very protective against severe disease. And there's a huge reduction in the number of associated deaths. So that was a very good aspect of the pandemic. The world was able to develop vaccines, effective vaccines, quickly. There were still huge problems with equity and delivery of vaccines in an equitable way. So it was very much richer countries having more doses than lower and middle income countries, which was is probably one of the biggest failures of the pandemic. And so overall, while I mentioned that there were positives with vaccine with the pandemic, overall I think the world didn't do very well. We were pretty reliant on sort of rudimentary measures like just not going outside. We weren't prepared in a way that minimized the impact of the pandemic. It caused huge suffering. And so when these things happen, people get together, the reports were authored by scientific, governmental, and industry experts. There are a number of recommendations for how we, as a world could do better to enable readiness of particularly diagnostics, therapeutics, and vaccines for the next pandemic. And one of the approaches is this idea of producing a library of prototype vaccines. So how can we better prepare for pandemics? We know that there are 25 different viral families known to infect people, or at least known to contain viruses which infect people. Now, whatever causes the next sort of viral pandemic is very likely to come from one of these viral families. Even though it might be a brand new disease like SARS-CoV-2 was, it was also a coronavirus, and we had some experience with coronaviruses, so it wasn't a complete unknown. And so the idea is that if we can develop vaccines against viruses in each of these viral families, then whatever does cause the next pandemic, hopefully we will have a prototype of a vaccine that we've already developed for a similar virus, and then we can transfer some of the learnings that went into making that virus to better respond to the new virus. Basically, as developing vaccines against MERS sort of helped us against SARS-CoV-2, we'd already known about the spike protein as the obvious target. We'd had some experience of what protective immunity entails. And so that's essentially the plan, to develop vaccines against all of these different viruses so that we'll have one that's very closely related. And so that's the sort of final program I want to talk about. We're doing that now for New World arenaviruses, the viral family. And so arenaviruses are broadly split between Old World and New World. Old World so Old World has things like Lassa fever in West Africa. New World affects the Americas. And there are 5 different viruses known to cause human disease in South America. Junin is one of them, which causes Argentine hemorrhagic fever. It's a very high case fatality rate. It's spread by this mouse, and the endemic range is increasing throughout the years, and that's through some of the standard reasons for these increases in endemic range. So climate change, urbanization, and so it's a problem, and it's becoming more of a problem. And basically, how do we make a vaccine against this, and what can we learn about making a vaccine against this that we could apply to other New World arenaviruses? So this is the schematic of the virus. What we really want to target is this glycoprotein at the top. So this is called the glycoprotein precursor. And again, this is the target of neutralizing antibodies. If we can prevent infection, then we can prevent all the downstream severe disease consequences. And so for this approach, we're taking a kind of a comprehensive approach to the antigen design phase. So this is what that glycoprotein looks like broadly, if it was a cartoon. So it's a trimeric structure. And each part of the trimer is composed of a sort of heterotrimer of GP1, GP2, and a stable signal peptide. So part of this project was, what can we do to that antigen? If we select that antigen, we think that's the bit that we really want to develop protective immunity against. Is there anything we can do to it that would enhance its immunogenicity? And so we make vaccines which are just wild type. We work with structural biologists to try and stabilize this GP1, GP2 formation in the pre-fusion formation. We looked at, can we use different cleavage sites to get better, more homogeneous cleavage? Can we get rid of glycosylation sites? Can we use different linkers, perhaps make a secreted version? And so the upshot of all of this whole vaccine program was we had 12 separate designs of this single antigen. And then we put those designs in 2 different platforms, ChAdOx1 and mRNA. And so if you think back to that sort of the vaccine development pipeline, it's relatively cheap on the left-hand side when you're talking about antigen design, and it's incredibly expensive when you get over to the right-hand side of phase 3 testing and GMP manufacture. So while it's relatively cheap, it means that we can make 24 different vaccines essentially for the same thing, and then we can test them in mice and see if it's immunogenic. I'm going to skip over this bit. So the project essentially involves these 24 different vaccines. We're looking at firstly testing these in mice, and then based on the immunogenicity, we'll down select the best 6, and then we'll go into an established guinea pig challenge model where we can look at efficacy and see how they actually work. And then based on that, as long as there are hopefully at least 2, then we can then go into potentially NHP efficacy studies, but also importantly, phase 1 clinical trials. And so the overall project, the overall aims of this project, well, one, can we design and develop a Hoonian vaccine? Because that would be very beneficial in and of itself. But more broadly, what can we learn about the process of making a Hoonian vaccine that could contribute to the sort of playbook for New World arenavirus vaccine development? So what can we learn about antigen design? What can we learn about what protective immunity looks like that may be useful if there was an outbreak of another New World arenavirus? And so this is the very busy slide, but I just want to convey that when you have these 24 different vaccines and you test them, you test immunogenicity, you end up with a lot of data. And this is just with three separate assays. So a PNA, the pseudo-neutralization assay, an ELISA, interferon gamma ELISpot. And if you rank the 12 different vaccines based on the different assays, firstly, you've got a post-prime ranking, you've got a post-boost ranking, you've got one of your vaccine platforms, the other vaccine platform, and you might see some sort of common things which come out. So you might say, okay, constructs 1 and 8 are pretty good at neutralizing across the board. Construct 10 is pretty good at binding antibodies. ELISpots, it's another construct. But how do you distill all of this kind of mess of immunogenicity into what are your best constructs? Because we need to select 6 of these and then take them into guinea pigs. So doing that is very simple when you've got a known correlate of protection, but it's very difficult when you don't know that much about what protective immunity actually looks like. So you need to go back to the literature and think, well, what is known about this Hoonian virus? What happens when people are naturally infected with it? What sort of immune response do they generate? Is there anything known about people who get severe disease versus people who just get mild disease? How can we apply that to come up with a system where we're confident that the immunogenicity that we're seeing is the sort of the immunogenicity that we really want? And partly, without knowing a correlate of protection, you're not going to be 100% confident that you're taking the best constructs forward. But then when you test them in the animals and you see efficacy, then you can start bringing it back and saying, well, okay, we've taken these constructs forward. We've looked at what the immune response is in guinea pigs for these different vaccines. These ones survived, these ones didn't. So what was different about their immune response? And then you can go back and get a better idea as you understand what correlates of protection are. So just to finalize, I've sort of explained about some different vaccine programs that are ongoing at different stages of outbreaks. What we're ultimately trying to do is, through all of these vaccine programs, whether it's preclinical or clinical, is progress the vaccines in a way that they can progress from preclinical to phase 1 to phase 2 to phase 3 and eventual licensure, but at the same time try and understand what's going on immunologically so that we're in a better position to understand why it is that they either do work or they don't work. And so we have access, as the global scientific community, to really powerful technology now that's able to really look in depth at the sort of immunology and what's going on post-vaccine. So we've got sort of system serology where we can look at multiple different antibody types for multiple different antigens at the same time. There's flow cytometry where you can look at 70 plus markers and get a really in-depth idea of what's going on in the cellular side of things. It's fine needle aspirates, we can look into lymph nodes, find out what's going on, and also things like tissue on a chip, looking at sort of tonsil organoid models, but not forgetting some of the basic and in many ways still most informative sort of immunology. And that is the basic assays of ELISAs and ELISpots and neutralization assays. And doing all this together to get progress the vaccines, but in a way that we really understand why it is that they're working or not working, and using that knowledge in new vaccine programs to help guide us in developing vaccines which hopefully prove to be efficacious. So I'll leave it there. Just that it's a huge number of collaborators and everything with all these different vaccine programs, particularly the clinical trial volunteers. So none of the clinical data is possible without them. But yeah, thank you very much. I'm happy to take any questions.
Yes, thank you, Danny, for such a wonderful presentation. I will start from the questions on the chat. There is one from Patience. No, it's from Nachema, asking, in your pregnant animal studies, did vaccination show any adverse effects on the fetus, and was passive immunity transferred to the offspring? Also, did you include a licensed commercial RVF vaccine as a control?
Good questions. So with that study, all the animals were autopsied within a couple of weeks. So we didn't look at seeing whether we didn't bring the animals to term. So we didn't look at whether the fetuses or the newborns had passive immunity from vaccinating the pregnant mothers. So we didn't look at that. We also, in terms of a commercial vaccine, so the most widely used commercial one is as I mentioned previously, the live attenuated one, so it actually can't be given to pregnant animals. So we didn't have a comparative vaccine that we gave to pregnant animals during that study. There are some vaccines now which are more attenuated and considered safer for pregnant animals. But there's still a massive need for a better livestock vaccine.
Thank you. There is also another question from Marianne. How often does mutation occur in the RVF gene TC glycoprotein? Could the JADOX RVF vaccine work for mutant RVF strains? Have you also tested whether this candidate includes generation of non-neutralizing antibodies that confer protection through ADCC?
Yeah, a really good question. So we are very fortunate in many ways with RVF that there is actually only a single serotype, at least that we know of. So the antibodies that we're generating with this vaccine will work against any of the isolates that have currently been isolated. Whether that continues to be the case, we still need to. I think it's always important to monitor the situation, especially if you roll out a highly effective vaccine. Then that introduces a new selection pressure where you might be sort of in some ways encouraging sort of mutants to develop. But we're fortunate with RVF that there is at the moment only a single serotype, so it bodes very well for vaccines. We're not particularly worried at the moment about sort of trying to develop a vaccine which is cross-strain protective, because we're confident that we do have a cross-strain protective vaccine. There was another part of the question, I've forgotten it, sorry. The last.
Is, the second one. Have you also tested whether this candidate vaccine induces
the non-neutralizing antibody? Yeah, so we've looked at binding antibodies, and we've looked at and we've looked at neutralizing antibodies. There is a significant correlation with neutralizing antibodies and the binding antibodies by ELISA, but we haven't looked at sort of FC functions and ADCC in that. But it's with RVF, because there's such a strong association with neutralizing antibodies and protection. That tends to be what people focus on, for good reason. But there absolutely might be other aspects of protective immunity that we'd like to look into.
Thank you. There is also another question from Jose. For the RVF trial, given that it was a single dose schedule, did you look at longevity and maturations of the antibody response?
So far, we've looked at, in terms of immunogenicity, it's simply been binding antibodies towards the two glycoproteins, looked at neutralization, and we've looked at the interferon gamma ELISpot response. We are intending to look at a much larger array of things. So the ongoing, what we currently have is the relatively small phase 1 studies in the Oxford and Uganda. much larger trial in Kenya is ongoing, and there'll be a study again later on with more people. So when we have these much larger sample sets of PBMCs and serum, then yeah, absolutely, we'd like to look more in depth at a lot of different things.
Thank you. The AstraZeneca vaccine was withdrawn. There is one from Fasan. The AstraZeneca vaccine was withdrawn for commercial reasons, but TTS played an unfair role. Will Rift Valley be similarly affected?
So I think I might have a slide on the bit. Yeah, I did. So yeah, absolutely. It's this bit came out. Oh, sorry, have I shared that? Can you see? Can you see this?
It's,
Oh, sorry, I'm in it. Wait, hang on. There we are. Right. So these very rare but very severe cases of what's known as VIT, so thrombosis with thrombocytopenia. This came about. This was identified during the rollout of the Oxford AstraZeneca vaccine, and it wasn't possible to see these sorts of adverse events in clinical trials, even very large clinical trials. You know, there were some 30,000 people trials for this vaccine in COVID. It doesn't come out that because it was too rare to see. But when you roll out a vaccine to millions and millions of people, these very rare effects can be identified. And we still don't know exactly what causes it. It's something to do with induction of antibodies towards platelet factor 4. But why the vast majority of people don't induce these antibodies, and some people do, is not well understood. But a lot of people are trying to understand this, particularly preclinically, because it's difficult to see clinically because you need to vaccinate millions of people before you'll start to see it. But a lot of people are working on identifying the mechanism of action. One thing that's clear is there's very different rates depending on geography. And so the Nordic countries, Australia, UK, were comparatively much higher than some other countries. And not all of that is based on the healthcare and surveillance infrastructure. So countries like South Korea, who are very strong surveillance infrastructure, saw almost no cases. And so it's not well understood, if it's some genetic background or why it is that certain areas saw much higher cases than others. It was seen with not just this vaccine, but with other viral vector vaccines. So it may be something that is common to viral vector vaccines. So it's possible that any viral vector vaccine, so including Challeps 1 RVF, this could be a potential issue. I think it's important to point out that any therapeutic, any vaccine, it's about what is the risk of taking it and what is the benefit of taking it. And for something like RVF, it's not a vaccine that would be widely rolled out to everyone in Africa where it's endemic, when only a small portion of those are actually likely to become exposed to RVF. It's a vaccine that will be used in response to an outbreak, and perhaps those that are the most at risk. And that changes the risk-benefit ratio at all. So you wouldn't give it to someone that doesn't have much of a chance of getting RVF. But during an RVF outbreak, if you work in a slaughterhouse worker, then absolutely the risk is comparatively so much higher that you can get RVF, and given the severity of the disease, that actually it's likely to be very much in the vaccine's favor to use in those situations. So it depends on the disease severity and how likely people are to get it. But it's still an issue, and it's still being investigated. And hopefully, if it's the mechanism is identified, then it can be modified in some way to get rid of the VIT. That would be ideal.
Thank you. I'm on the Q&A section now. There is a question from Komal asking, I want to know, is there a waning antibody-mediated protection during COVID-19 vaccination, which are based on spike antigen? how important is the COVID-19 vaccines, which are based on other antigens, mostly non-structural proteins for long-term protection?
It's a good question. The short answer is I don't know. It depends on the antigen that was in the vaccine, and then the circulating antigens. I know, at least in the UK, the people who are receiving yearly boosters are older people. The rest of the population are not receiving any vaccines, and they've updated their vaccines at least once or twice. I think the current one that's given to elderly people here is a sort of bivalent vaccine that contains different variants of concern or different Omicron strains. So I don't have any data to know to sort of demonstrate that how effective they are in the long term. I think from what we know from the beginning of the pandemic, as the virus was evolving, the vaccines were becoming less and less effective at preventing infection, but they still remain very effective at reducing severe disease. And ultimately, that's what we want vaccines to do. It's great if they can prevent infection, but what's really important is, can they prevent severe disease? And they were doing that very well.
Thank you. There is a question, more of a comment and a question from Mwati. Congratulations for RVF vaccine being approved for human use. Are there strategies to actually test field efficacy during an outbreak?
Yeah, so actually this very hot off the press is that the Channels 1 RVF vaccine was very recently licensed to Serum Institute of India, and they have produced 100,000 or are producing 100,000 doses of this vaccine, 10,000 of which are apportioned for a clinical trial looking at efficacy. And the idea was to move as quickly as we can and do a clinical trial in the Gambia for the ongoing outbreak. It looks like that outbreak will peter out on its own, which is great news. But it goes to show that even though this has been quite quick to, well, it's been a slow process doing all of the trials, but that's fairly common. But when an outbreak has occurred, because we had that phase 1 efficacy data, phase 1 safety and immunogenicity data, and given the strengths of the platform that it can be manufactured quickly and at low cost, that we're able to move quite quickly and form a partnership or license it to the Serum Institute and be in a position to have 100,000 doses ready to test. But even that wasn't quick enough for this outbreak. But we're in a much better position now that during the next outbreak, the Serum Institute will have finished these vials, and they could be tested much quicker. But you really do need to be so quick for these outbreaks in order to have a vaccine in the country and ready to be used and set up a clinical trial. It's very difficult to be quick enough that you're able to actually test efficacy. But we're in a much better position now. We've got the vaccine licensed, and Serum Institute are making a large number of doses.
Thank you. There is also a question related to the 100-day mission. Overall, from Najema, overall, as a vaccine development expert, what do you see as the biggest scientific bottleneck preventing a true 100-day vaccine from sequence to deployment, excluding market demand and outbreak-driven acceleration?
Yeah, it's a really good question. I think what is most difficult, and perhaps securing the funding is one of these things. I've mentioned how this is much easier now in this, particularly post Ebola, post COVID, where it's much more at the forefront of funders and the general public's sort of thoughts, I guess, that we do need to be developing vaccines quickly against these outbreak pathogens. I think one of the biggest disappointments and perhaps challenges is that it seems to get forgotten quite quickly. So even post-COVID, there was a lot of promises of all these, setting up these different manufacturing hubs. And there's a lot of good that came out of it, but a lot of the sort of will about, we need to be much more ready the next time. We need to have these manufacturing hubs. We need to set everything up and have it kind of rolling along. But the second an outbreak comes, we can sort of switch things on and get going quickly. actually that seems to get forgotten quite quickly, and it doesn't take very long to go back to the old ways and to think, actually, how much has changed since COVID? We certainly, in terms of vaccines, we've gained a vast amount of knowledge of how to make vaccines quickly, but I still fear that it's fundamentally, are we in a completely different position now than we were pre-COVID? I don't actually know that I don't know that we'd be that much quicker responding to an outbreak now. It depends on the pathogen, but so I think staying focused on funding and a will to produce these vaccines quickly is a challenge.
Thank you. Funding will definitely be a challenge because of some funding withdrawals. Yes. Yeah. Now there is also another question from Jose Campos. One of the potential withdrawals of viral vectored vaccines is the development of vector antibodies. Do you think there is a risk that using JADOX1 across many pathogens could compromise its use for pandemic preparedness in the long run?
Yeah, another very good question. I think I've got, so if, here we are. So yeah, that's a really good question. So one of the main reason we use a chimpanzee adenovirus rather than a human adenovirus is because we want to avoid anti-vector immunity. The way this vaccine works is as a delivery vehicle to take the gene inside our cells. If our immunity is sort of stopping that, then we're likely to see a reduced response from the vaccine. So now that we have, apparently there's almost 3 billion doses of ChAdOx1 COVID been administered, then these people are likely to have some form of anti-vector immunity. So we do now have some data on what the impact of that is. The first comes from COVID. So we had, just get my pointer. We had a first dose here, and then when they were given a second dose, and at the time we didn't even know that a second dose would be, how effective that would be given the immunogenicity, the anti-vector immunogenicity elicited after the first dose. But it turns out you give a second dose and you have a much improved antibody response. And then here, this line here is about 9 months later after the second dose, when people were given a third dose of ChAdOx1 COVID, antibody responses did peak. So they peaked back up to, well, even potentially slightly higher. So we know that we can even give 3 doses, and we're still not seeing enough anti-vector immunogenicity to really dampen the response. The second bit of sort of evidence comes from, at least initially, this is looking at the response to SARS-CoV-2 in participants that had prior ChAdOx1 vaccines. They're enrolled in a previous ChAdOx1 trial or no prior ChAdOx. And we can see that actually we do, at least initially, the early response in purple is slightly higher in people that didn't have previous ChAdOx. But when they're given a second dose, it tends to get smaller and smaller, and it wasn't significant at this stage. We've since done a number of clinical trials based on new ChAdOx1 vaccines to different diseases, where we've stratified the participants between those that received 2 ChAdOx doses during COVID and those that received alternatives like mRNA vaccines. And again, we see something similar to this, where we do see some drop-off in immunogenicity. It's not significant in people who had never had ChAdOx before, or rather, we see a drop-off in people that did have 2 ChAdOxes before, but it's not huge. So the first sort of takeaway is that it's not a catastrophe in that we've given, people have received 2 doses of ChAdOx, therefore they can't receive another vaccine. It clearly is possible to have an immunogenic response to your 3rd or 4th dose of ChAdOx vaccine. It's slightly more complicated than just stratifying participants between those that received ChAdOx vaccines previously and those that didn't, because at least in the UK, those that received ChAdOx tended to be older. Whereas the younger people had the alternatives. So there are some confounders in, is it anti-vector response from people because they've had 2 vaccines, or is it because they're a bit older and that's why they have a slightly reduced response? But we don't see a huge drop-off. Essentially, there is potentially some impact, but it seems like you can still give ChAdOx1 vaccines and they can still be immunogenic.
Thank you. There is another question on the chat from Sharon. what variants of the antigen do you use in making the vaccine prototypes in preparation for any potential pandemics?
Yeah, that's a really good question. So in terms of being prepared for developing a sort of a broadly protective vaccine, I think understanding firstly how to develop a broadly protective vaccine, and that's not necessarily easy, but understanding which parts of the pathogen are conserved and which parts of the pathogen you can target and that elicits protective immunity is very important. So understanding how diverse viruses within a family are is sort of crucial to understanding how likely it is that you're able to provide or able to produce a vaccine which is going to protect against multiple different strains. The other thing that, if you can have a cross-protective vaccine, that's the ideal situation where you have a single vaccine and it protects against multiple strains, even if they don't yet exist. The other thing is with these vaccine platforms is that they can quite quickly, you can switch out the genes. So it's possible to update the vaccine, and that's what's happened with COVID, where the vaccines have been updated so that as they became less effective of the new circulating strains, you update them and then they become more effective at what's currently circulating. So I think there's a lot of work to do on the immunogenicity and what constitutes a broadly protective immune response, and trying to develop vaccines which elicit a response towards more conserved regions, which will then protect. So that's a couple of different sort of strategies that you could employ in it, but it's a big challenge depending on the virus.
Thank you. There is another question and comment from Welty Okete on the Q&A section. Thank you for the insightful talk. With the growing popularity of vaccines, is it a future plan to integrate RVF into one of these platforms? Are there general factors that determine the suitability of a vaccine or a pathogen for vaccine platforms?
Yeah. So was that about using the RVF sort of antigen in different vaccine platforms? Did I understand that correctly?
It's on the Q&A section.
Sorry, could you repeat that?
Yes.
I'm not sure I understood the actual.
With the growing popularity of vaccines, is it a future plan to integrate RVF into one of these platforms? Okay. There are general factors that determine the suitability of a vaccine or pathogen for vaccine platforms. Yes.
Yes. So I think this is a particular problem or a potential problem for a lot of these pathogens, is that RVF outbreaks are extremely sporadic. So particularly when it comes to veterinary vaccines, in order to make it as sort of cost effective and as beneficial as possible, if you could then combine sort of an RVF vaccine with another vaccine that protects against a different disease but perhaps is more common, then that hugely increases the sort of utility of an RVF vaccine, because then you're not trying to convince farmers, for example, that to spend money on this disease which maybe they've not heard of, or maybe it only happens every 10 years. But if you can couple it with a vaccine that they very much do need, they use a lot, then you're automatically making the vaccine a lot more useful. And I think whenever you combine these vaccines, it's you need to show that you're not having an adverse effect on the original vaccine. You need to show that they both you can administer them both, and you still get that that good response that you would expect if you administer them individually. But you can massively reduce costs by pairing vaccines or using multiple vaccines at the same time. I think that's what we do in humans and animals anyway. So there's huge incentive to do that, and I think it's absolutely important to look into how you can, when you're actually rolling out these vaccines, how can you combine them to other vaccines, and so that it's efficient and cost effective.
Thank you. There are no further questions on the chat or the Q&A section. Give 1 minute to 2 minutes. If there's anyone with a question, you can use the icon, raise your hand. and Danny will answer the question.
I'd probably just like to add a bit about the talk about the biggest challenges with the question for this 100 days mission. I think actually I've thought about one of the biggest challenges actually getting the vaccine to where it's needed, and COVID was an example where that didn't work well on a global scale, right? So the rich countries tended to buy huge stocks, and it was the less rich countries which were far less well served. And there are people in some countries that are on their sort of third dose of vaccine, and then other much more vulnerable people in other countries that are only having only able to get one vaccine. So I think some of the politics around how the vaccines are sort of distributed is going to be one of the biggest challenges, because it's not just about having an effective vaccine. It needs to be actually administered to people before it's of any use, and that part, getting it to where it needs to be in a fair way, is, I think, a big challenge.
Sure, it is, especially in the global south. There is a question from Atta. Atta, can you hear us? Can you go ahead?
Okay. Thank you. So this question is a bit different from the vaccine platforms that have been discussed by Daniel. I just wanted to ask to know if peptide vaccines have a future in the development of vaccines against outbreak pathogens.
Yeah, I mean, there are so many different, but I know I've definitely overly focused on viral vectors and mRNA because that's what I'm most involved with. But there are a huge number of different vaccine platforms, and they all have strengths and weaknesses. And it's no different for peptide vaccines. I think it depends on the type of immune response you're wanting to elicit. So peptide vaccines might be particularly good for those pathogens which require a good T cell response. It depends on the peptide, but a lot of the antibodies will respond to or will bind to conformational epitopes, which you might be unlikely to get or less likely to get from a peptide, where you're sort of stuck with linear epitopes. But for those pathogens which do require a cellular response, and for all of these, the immune system is complicated and it works in concert together. So we sometimes oversimplify things by saying, this needs a humoral response, this is a cellular response. It's all working together, and I think peptide vaccines absolutely could have an important role to play if they're shown to be effective for a particular disease. And it's basically there is no one type of vaccine which is the best one. You can't rank them as from best to worst. They all have their individual benefits and drawbacks, and it's not just about safety and efficacy, as important as they are, but it's the other things. It's how much they are, how the cost of producing them. It's how manufacturable they are, how stable they are, and all of these different platforms, including peptides, have their different strengths and weaknesses. So it's I think all of these vaccine types have a role to play.
Sure. There is no question on the Q&A or the chat box. Maybe Dan, you could make your final remarks as we close the webinar. Thank you.
Okay, so thank you very much. I would say when it comes to making vaccines against outbreak pathogens, there are a lot of the development pipeline of making vaccines is relevant for whether it's an outbreak pathogen or whether it's not, whether it's malaria or TB or HIV. A lot of it is the same process. But certainly some of the aspects which are particularly important or relevant for outbreak pathogens is speed of making the vaccine, speed of testing the vaccine, and I think some of the difficulties with outbreak pathogens are in the licensure, because it's not necessarily easy to test for efficacy. So those other sort of pathways towards licensure, like the animal rule, it really requires understanding of the immune response. It's not, if you can't demonstrate efficacy just empirically, then and you need to say, well, this is what's going on in protected animals, this is what the immune response looks like in humans. You really need to understand that immune response and be able to come up with a convincing sort of argument as to why, in the absence of direct efficacy results, what you've got in humans is going to be a protective vaccine, and that requires a lot of these other sort of exploratory immunogenicity studies around vaccines. So yeah, that's what I'd say.
Thank you, Danny, and thank you, everyone. In this webinar, we would like to hear about your experiences and what else you would like us to discuss in upcoming forums. Before we close, kindly requesting that take a few minutes to respond to the post-webinar survey posted in the chat. There is a link on the chat box. Have a nice day, and looking forward to more engagements with you. Thank you.
Thanks so much all. Bye-bye.